Welcome to the Stanford Medicine Alumni Association's Alumni to Alumni Speaker Series. My name is Jennifer Cobble, and I'm the Executive Director of SMAA. This series features accomplished graduates of the Stanford School of Medicine as they share their knowledge and experience in a variety of healthcare, research, startup, and private sector settings. With these presentations, we hope you'll gain a better understanding of the diversity, innovation, dedication, and impact of the many alumni who represent the Stanford medicine community as leaders in both medicine and the biosciences. Before we begin, I'd like to remind you that your microphones and visual profiles have been set to listen-only mode. Only the speaker and the moderator will be on your shared video screen. Should you encounter technical difficulties, please try leaving the webinar and logging in again. And if issues persist, you can reach out to us at medalumni@stanford.edu. If you'd like to make your slides a little larger on the screen or have Dr. Maldonado look larger on your screen, you can put your cursor between those two images and move from left to right to enlarge or shrink. Finally, we'll be answering questions at the end of the presentations. You can submit your questions anytime during the talk by using the Q&A feature at the bottom of your screen. If you see a question similar to your own, you can like it by clicking on the thumbs up icon, and that question will move up in the priorities. But given our large audience, I regret that we cannot ensure that all questions will be answered in the time that we have allotted. Now, I'm pleased to begin our session, Curing COVID, the Outlook for Vaccines and Therapeutics, with Dr. Yvonne Maldonado. Dr. Maldonado has been on the forefront of COVID-19 efforts at Stanford Medicine, as well as contributing her expertise to government policy both on the state and national levels. She's been closely involved in Stanford's research on tracking, testing, treating, and curing COVID-19 patients and identifying strategies to address global health in a pandemic. She is the Tobey Professor of Global Health and Infectious Diseases and Professor of Pediatric and Epidemiology and Population Health at Stanford University. She founded Stanford's Pediatric HIV Clinic and serves as chief of the Division of Pediatric Infectious Diseases at Lucile Packard Children's Hospital. In addition, she's an advocate for diversity, equity, and inclusion through her role as Stanford Medicine's Senior Associate Dean of Faculty Development and Diversity. Dr. Maldonado has contributed her medical expertise to government policy, serving on the National Vaccine Advisory Committee through the United States Department of Health and Human Services, and as a member of Governor Newsom's California Initiative to Advance Precision Medicine Advisories Council. She's the chair of the Committee on Infectious Diseases at the American Academy of Pediatrics and serves as a senior fellow at the Stanford Center for Innovation in Global Health. Her research focuses on the epidemiology and prevention of infectious diseases such as rotavirus, measles, mumps, rubella, polio, and pediatric HIV infection. Her research program also focuses on the development and the implementation of vaccines. Most recently, she and her team have been involved in many of the studies at Stanford, seeking to understand the biology and the epidemiology of COVID-19. Raised in Los Angeles, Bonnie's the daughter of Mexican immigrants and was the first in her family to attend college. She received her bachelor's degree from the University of California, Los Angeles, and then attended Stanford University School of Medicine, where she received her MD degree in 1981. She then performed a residency in pediatrics at Johns Hopkins Hospital, where she remained for her fellowship in pediatric infectious diseases. In 2018, the Stanford Medicine Association was pleased to present her with the Rise Award for Outstanding Lifetime Contributions to the Stanford medicine community. Please welcome Dr. Yvonne Maldonado.
Thank you, Jennifer, for that introduction. And I want to make sure that I can access the slides. Well, welcome to everybody, and thank you for joining me today. I do want to give you a quick, but hopefully detailed enough overview of the outlook for vaccines and therapeutics around COVID-19 and SARS-CoV-2 virus. And hopefully we'll have some time to answer questions as well. So let's talk a little bit about the virus so that we can understand some of the vaccine platforms first. First of all, you probably all know, but it really helps to start from the beginning, that SARS-CoV-2 is a member of the coronavirus family. And we know that there are 7 human coronaviruses, 4 of which are seasonal viruses and cause about 30% of all colds, seasonal colds, around the world. 3 of the additional coronaviruses actually mutated from animal reservoirs and have become human viruses as well. This includes SARS, which showed up between 2002 and 2004, the Middle Eastern Respiratory Virus and MERS virus in the mid-2000s, and then now, of course, SARS-CoV-2. They all, by the way, came from bats. So we know that that was the original reservoir, and what the intermediate host is really hasn't been clear yet. Now, what's really interesting and helpful, fortunately for us, about this virus, although it is fatal, it produces fatal illness, as we know, it's not anywhere near as fatal as the original SARS virus or the MERS virus, but it is more prevalent. So the original SARS virus is basically gone. We don't know how it left, and we don't know if it'll return. And MERS is still around, but it is easily suppressed by reducing contact with camel secretions. And so this particular virus is notable because the spike protein, as you can see here in this cartoon, is really the main determinant in neutralizing immunity. So if you make antibodies to these spike proteins, these are on the surface, it forms a conformational, basically trimer, 3-part spike that actually attaches to the ACE2 on the surface of the human cell. And by attaching, will then allow for cell entry of the viral RNA. And because of this relatively simple way of neutralizing infection, we have been able to develop vaccines against that spike protein that really do neutralize the ability of the virus to infect human cells. And so if you look here, there are just a list. This is not meant for you to read, but just to see that just in the last month alone, we've seen numerous changes around vaccine developments around here in the US and around the world. And I'm going to talk specifically about 2 of the vaccines because they are novel platform vaccines, and they're also the ones that are licensed in the US. But just so you see that there's constant movement and activity around the many different vaccine candidates around the world. So my favorite go-to place for good graphics lately has been the New York Times, and they have a really nice coronavirus vaccine tracker that gives you just an overview of what the different vaccines are and which ones are in use. So it shows you here that 3 have been abandoned and 2 are currently authorized, have expanded emergency use authorization. And in this country, as you may know, we have 2 vaccines that are under EUA from the FDA, and that is the Pfizer BioNTech vaccine and the Moderna vaccine. And there are 3 other vaccines, Johnson & Johnson, the Oxford AstraZeneca vaccine, and Novavax vaccine, that are being considered by the FDA in the near future. Hopefully the Johnson & Johnson will be considered next month, we hope. We should be seeing that data. I work on the ACIP, the CDC ACIP committee, and we'll hope to be seeing that soon. Now, what is interesting about the Moderna and the Pfizer vaccines is that they are based on well understood mRNA biology. And what many of you may know is that mRNA is the blueprint for all protein synthesis. It uses cell biology basically to activate the immune system. So there are inherent safety features of this mRNA. It's basically a synthetic messenger RNA. Now, in the normal human cell, mRNA is made in the nucleus and then is transported out into the cytoplasm, where it basically serves as a template for protein synthesis. But in this case, what happens is you get mRNA as a template being injected. So it doesn't come in from the nucleus, it doesn't enter the nucleus. It comes straight from the vaccine into your myocytes and then in the deltoid muscle. And then at that point, it enters the cell. And the manufacturing process itself is cell-free, and contains no human or animal products, preservatives, or adjuvants. So it's quite relatively straightforward. And of course, we can say that, but it is a real breakthrough scientifically to use mRNA. And in fact, no mRNA vaccine had ever been licensed before. But we do know that, sorry, I'm trying to make my slides move here. There we go. So what happens is you inject the mRNA. It's surrounded by a lipid membrane, and that lipid then helps preserve the mRNA while it's being injected. And then you have mRNA that serves as a template in the cell. You get immediate translation into spike protein. So it's just synthetic RNA that is codes for the spike protein. You get lots of spike protein, and that mRNA immediately disintegrates. Within minutes to an hour or so, the mRNA is gone. So it disintegrates, it doesn't integrate, it doesn't get into your systemic circulation. It's just gone, and the fat, the lipid as well. And then those spike proteins are expressed on the cell surface of that infected cell, and then you develop immunity to that. So very high levels of antibody are developed in this way. And you can see here that for the Pfizer vaccine, and very similar for the Moderna vaccine, is these are indicated for prevention of coronavirus disease, not infection, but disease. And there are different dosing for the two different products, but the storage is actually quite cold, -70 for the Pfizer and -20 for the Moderna. Now, the indications that I mentioned in the studies that were done were for symptomatic disease. And you can see here, these were people who came in with symptoms, a whole degree of symptoms, plus a positive PCR. So we did, the studies were not done because of, in order to do these rapidly, they did not look for asymptomatic infection or shedding. They looked just for symptomatic disease. And a summary of those slides is here, of those studies is here. So you see that almost 75,000 people were involved in both of those studies. And the primary endpoint was similar, that is PCR positive with COVID symptoms. And there were a total of 162 placebo recipients who developed disease for the Pfizer trial, as well as 185 placebo recipients in the Moderna trial, and then a total of 19 vaccinees for both trials who developed symptomatic disease, with vaccine efficacy between 94 and 95%, with a lower 95% confidence interval of about 90%. So highly, highly efficacious vaccines. And the endpoints were different: 7 days for Pfizer and of the second dose, and 14 days after the second dose for the Moderna vaccine. Now, there are some overall side effects. I just saw them this morning. I was on a 6-hour ACIP call, but I didn't have time to put the slide up. But basically, overall adverse events, those of you who've been vaccinated have experienced some of these, but they are quite frequent. Over 50% of people will report some adverse events. Most of these are local site tenderness and some flu-like illness, headaches, and myalgias. The vast majority of those are gone within 48 hours of vaccination. And there have been cases of angioedema noted and anaphylaxis as well. And the anaphylaxis is for the Moderna vaccine has been noted to be about 2.5 cases per 1 million doses given. Interestingly enough, for this vaccine, it's all been women who've had anaphylaxis, and 80% of all of the anaphylaxis cases have occurred in people who have allergies to food or other products. The Pfizer vaccine has have reported anaphylaxis of about 11.1 cases per 100 per million doses. And for comparison's sake, the flu vaccines given produce about 1.3 cases of anaphylaxis per million doses. So we know that it happens, but these have all been treated in the outpatient setting and the individuals have fully recovered. We did pause in California the Moderna vaccine for a cluster of less than 10 vaccine reactions that were reviewed by me and others on the California Vaccine Safety Group. There were no other similar reactions anywhere in California or the rest of the US. There was a specific lot involved, and this is a large community-based drive-through vaccines clinic. And no hospitalizations or deaths resulted, and it was deemed that many of these were most likely angioedema. We had a panel of expert allergists and immunologists, and that pause was lifted last week, and we have not seen further problems. And you can see here, I wanted to just briefly talk about the variants. So in addition to vaccination, one of the other areas that we need to deal with, not only around transmission, but around vaccination is whether or not we are going to be having problems with variants. And as you probably know, viruses mutate on a regular basis, but I'll show you a picture in a bit. These particular viruses, for some reason, probably due to selective pressure, are not only randomly mutating across the 30,000 kilo nucleic acid genome, but they are focusing in specific areas. And there are areas that are focusing in the gene area, the gene that codes for this spike protein. And those mutants, there's concern that over time, those mutants could actually become escape mutants and escape the neutralization capacity of the current vaccine. So at this point, there is a CDC website that shows you how many variants have been detected in the US. Now, keep in mind that this represents sequencing of less than 1% of all the viruses detected in the US, but it is a reasonable way to start surveillance. And we have seen that overall, as of yesterday, there were about 195 of these identified in the US, the vast majority in California, Florida, and New York, probably reflecting our ability to really do most of the sequencing and submitting those for testing. And here are the different variants so that we know of, although I am sure there will be additional variants that occur over time. That's what viruses do. The ones that are circulating globally, the first one that was noticed, at least in the literature, was the one B117, which emerged in the United Kingdom with a large number of mutations. I believe there were 23 mutations. This virus has actually been shown in a preprint to spread more easily and quickly than the other variants. And there is preliminary evidence that it may cause more severe illness or increased risk of death. But that data, again, is preliminary from the UK. And it has not been really gone through additional peer review at this time. But we are vigilant about that issue. This variant was first detected in September of last year, and it's now highly prevalent in London and Southeast England, and has been seen in around the world. And actually, we've seen some, as you may have seen, in the US as well. The other variants that are important is the one from South Africa. It's called 1.351. And this was originally detected in early October. And it shares some mutations with the variant detected in the UK, but not all of them. And there have been cases outside of South Africa, but not detected in the United States so far. And then finally, there is a variant called P1 that has emerged from 4 travelers in Brazil who were identified in an airport at the Haneda Airport in Tokyo. And that variant is distinct as well and has not been detected in the US. But we also have some variants of interest or variants of concerns here in California and other US states. So there are variants growing up in our country. And here's a map, a genotypic map showing what I was talking about, where you see random mutations. This is the genome of the SARS-CoV-2 virus, and it's 30,000 base pairs. And you can see here is the S protein, the gene that codes for the S protein here, and here's the gene that codes for the nucleoprotein. You can see that there are mutations scattered across the genome, but there are really a clustered mutants here in the area of the receptor binding domain that is where the virus binds to the human ACE2 receptor. And it turns out that some of those mutations, but not all, actually stabilize that trimer so that it's more likely to attach and stay attached. So therefore, it's thought that that is what contributes to the increased potential for increased infectivity. Whether it is more virulent and what the mechanism of that is not clear at this time. And just for the Stanford spin here is that this is from Ben Pinsky and Tom Montine here in the Department of Pathology, who run the clinical laboratory. And you can see that there is an assay here in our laboratory to detect the UK, South Africa, Brazil, and California strains, and they are looking for these strains actively. And these are the mutations that they're targeting at this point. So we are actively seeking those in our patient populations and reporting those to the states and counties as we need to. So I'd like to move on and talk about the therapeutic pipeline. And just in general, I wanted to take a global perspective. And here's the link to this particular website that gives you an overview. There are at this point 815 active unique compounds in development, and that includes things like antivirals, vaccines, and therapeutics. By the way, I'm well, I'll talk about it in a bit, but I am on a DSMB. I forgot to mention that's not a conflict, but I'm a DSMB member for one of the NIH clinical trials groups. There are 349 clinical compounds and 466 that are preclinical. So again, this virus has not been around very long, probably a little more than a year. So there's a lot of development and a lot of preclinical work going on. And you can see here, if you look at the therapeutic pipeline, that the vast majority of the therapeutics, I'll tell you what these color codes mean in the next slide, but these are different types of treatments. They are mostly in the phase 1 and phase 2 realm. There are a few that have made it to phase 3 and some, of course, in the phase 4 or post-marketing phase. And then if you see the numerous targets that these treatments have been aimed at, hopefully you can see that the number 91 different therapeutics are targeted for the anti-inflammatory impact. And then there are COVID antibodies, vaccines, and others. But the vast majority of the products that are being developed at this point are in the anti-inflammatory, anticoagulant range, and the antibody range. Very few that are really targeted as antiviral, true direct antiviral treatments. And those are some of the antivirals we're actually using here at Stanford. We have an inpatient and an outpatient treatment unit. I co-direct the outpatient treatment unit, trials unit, and we are using 2 direct antivirals as well as some of the monoclonal antibodies. Now, if you look here at the distribution of these therapeutics, you can see that the vast majority here, over 70% of them, are brand new. But there's a good number that are redirected or repurposed. And the redirected and repurposed products are the ones that have had obviously the most success because a lot of the safety work had already been done for previous indications. And you can see here that remdesivir and some of the monoclonal antibodies are at the top of the list of these repurposed drugs. And then, so I wanted to end the overall discussion here around some of the efforts that are being done at the national level, national network level. And this is something called ACTIVE, or accelerating COVID-19 therapeutic interventions and vaccines, which is a public-private partnership to coordinate research and speed the development of the most promising therapeutics here in the United States. Now, this is coordinated by the Foundation for the NIH, and it is jointly conducted by NIH, BARDA, CDC, FDA, and the Department of Defense and the Department of Veterans Affairs. What used to be called Operation Warp Speed, which doesn't actually have a new name yet, but we're going to call it that for now, and the European Medicines Agency. So it's really quite a transdisciplinary approach to hopefully to rapid development of therapeutics, and includes members of academia, philanthropic organizations, and obviously industry partners. Now, the 5-prong approach really looks at Active 1 through 5, and the Active 1 involves platforms for immunomodulator rapid trials. Active 2 is outpatient monoclonal antibody trials. Active 3 is inpatient monoclonal antibody trials, and that's the DSMB that I'm a member of. And you can find all of these on the website here below. And Active 4, antithrombotics. And then Active 5 is the big effect trial, which is really what we call an adaptive multi-pronged trial, where you basically have a platform of remdesivir, and then you build in phase 2 trials with rapid phase 2 trials with different therapeutics. So you can rapidly try to move on to phase 3s if you find some activity. The fast track areas, in addition to these 5, include the preclinical working groups, therapeutics clinical working groups, the clinical trial capacity working group, and the vaccines working group. And we have members, Stanford members of all of these working groups. So I'd like to start to wind down here and say that just yesterday we got an announcement that the Regeneron company announced a trial that we've been involved with, has had some pretty exciting results, although still preliminary. And the Regeneron product is it's an antibody cocktail. It's a combination of 2 pretty selective spike protein monoclonals. One of them has more activity than the other, but the idea of using a dual cocktail is that you reduce the degree of mutations that can escape or escape mutants from this particular therapeutic. Now, one of the approaches has been to give these monoclonals to sick individuals. So these are not indicated in severe disease because there is a mortality signal when you give these to individuals, not this product, but the Lilly product, to individuals who are hospitalized on oxygen therapy or in the ICU. So those trials have been halted. The other approach has been to give the drugs to the monoclones to individuals who are hospitalized with less severe disease to see if you can stop progression of disease and also reduce time in hospital. Those trials are ongoing, and the cocktail has been given emergency use authorization to prevent progression of disease. But this result here has to do with outpatient treatment in a novel way, which is prophylaxis. So this trial so far has enrolled 409 individuals. And the idea was to give individuals, instead of a 1 or 2 hour infusion of a monoclone with a 1 hour safety waiting period, to actually just give a subcutaneous injection of the monoclonal antibody product into individuals who were household contacts of a known PCR positive individual. And so that trial has shown so far that out of the 409 people, about half of among roughly divided at 50% received placebo and 50% received the monoclone as prophylaxis. There were 0 cases of symptomatic transmission in the treatment recipients compared to the placebo recipients, and a 50% reduction in an asymptomatic disease in the treatment recipients as well compared to the placebo recipients, and also significant reductions in viral load in the shedding among the treatment recipients, as well as a much reduced time of viral shedding. And that is about 7 days for the treatment recipients compared to about a median of about 3 to 4 weeks for the placebo recipients. So this trial is continuing to accrue patients, but we are hopeful that this could be, in a sense, a mini vaccine until we can really roll out vaccine in significant numbers throughout the country. So very exciting information, and one of the drug studies that we're doing here at Stanford. So what does the future hold? Well, based on what we know about these viruses and just viral treatments in general, especially using HIV as a good indicator, it's likely that we're going to need multiple antivirals, and we may need combination therapy, including antivirals, immunomodulators, et cetera. We know, for example, that anticoagulants have also recently been shown in 3 different trials to reduce severity, disease, and mortality outcomes. So it's likely that we will need a cocktail approach to treatment of actual infection, as well as potentially looking at prevention with vaccines and monoclonal antibodies. Now, it's also important, as you've seen with the NIH approach and others around the world, that clinical trials must be flexible and adaptive. It is difficult, frankly. I've been doing these trials now for several months, and it's very hard. It's not hard to enroll people who are sick and hospitalized. Those are harder endpoints to measure, however, but it's very difficult to enroll people who are outpatients who have mild or moderate symptoms because, first of all, they may not see a benefit in getting a therapeutic for something that they perceive as kind of a flu-like illness. And the second thing is it's very hard to move people around when they're supposed to be in quarantine and also don't really feel well enough to drive. So we have been able to be successful in finishing one trial. We're in the middle of 5 more trials. We're almost done with our second trial. And we have, again, 4 others in process right now. So we also would love to be able to see development of biomarkers or other clinical indices to measure outcomes, because then we could target individuals and say, listen, you are at X or Y risk of progression of disease. And it might be easier to not only target for trials, but to target for actual treatment once we get indications. So this has been another issue, but it's likely that for at least the rest of this, most of this year, if not the whole year, we're going to continue to need non-pharmacologic interventions, that is masking, social distancing, hand hygiene, and other practices, because we didn't really have time to talk about it, but this issue of vaccine-induced herd immunity is still unclear. We don't know yet, and the studies were not set up to identify whether you can actually stop transmission of virus once a person is vaccinated, either to the vaccinee or from the vaccinee to another person. I suspect that we will see reduced transmission, but not cessation of transmission, both to the vaccinee and to others. So that means that we will need to use masking and distancing for at least some time until we get a better handle on the natural history that has been affected by vaccine use over time. And so with that, I'd like to stop, and I'm happy to hand it back over to Jennifer at this point.
Thank you. Thank you, Bonnie. There's so much new information and it's a moving target. I think that people like Dr. Maldonado have been at a fast sprint for a year now, not only doing research around these topics, but also trying to be reactive to the pandemic itself. And it's been extraordinary amount of work being done not only at Stanford, but at many health centers around the world. And we're really blessed to have somebody like Dr. Maldonado, who's been kind of in the thick of things, though she keeps smiling, but I know every day is another challenge. You did talk towards the end. I'm going to field a few of these questions and maybe group a few together in around certain topics that people have mentioned. Just in your last couple of slides, you were talking about this idea about vaccinated people, if they can still be carriers. There are a lot of questions about how long do you think it'll be before we know whether you're vaccinated and not a carrier? If I interact and I've been vaccinated, can I interact with people who've already had COVID and recovered from that? Can we kind of hang around together without doing all the preparations needed? When are we going to see this so-called herd immunity and feeling that we can let down some of our cautions about social distancing and mask wearing and seeing family and hugging the person across the table?
Yeah, that's a great question, and that's actually this is actually what I studied with polio virus, because I was involved for many years and still actually hope to get back to it as soon as I can in the next year. Polio transmission, in particular, right now, not only wild polio transmission, which has been reduced substantially, but also live vaccine polio transmission. So a lot of my work has been in this area of viral transmission and how do we measure it. We don't have an answer now. The trials were not set up to do that because they wanted to get them out as quickly as possible. And enrolling 75,000 people in studies really required that they do just stick to the symptomatic disease with the positive PCR. We and others are doing larger scale trials where we're following people over time, We have a household transmission study. There are a couple that are funded by CDC as well. They reported on some of those this morning. There have been a couple papers around that area. We're in the middle of writing our papers as well to see, but these are pre-vaccine. So we now need to follow these patients and then see if they get vaccinated. And in order to do that, what we're focusing on here, and CDC as well, is we have a very large cohort of over 3,000 healthcare workers, most of whom are now vaccinated here in the Bay Area. And we've been collecting monthly nasal swabs and blood samples from them, and hope to follow them as we move along and see if they develop symptoms, or even if we do prospective studies to look for asymptomatic development of immunity of disease, or shedding, or antibody. And the reason the antibody would be helpful is because if you develop actual disease, you're going to make antibody to other than the spike protein. If you get vaccinated, you only make antibody to the spike protein. So we can actually distinguish between infection and vaccine-induced immunity, which is really fortunate. So we won't know the answer to that for another few months yet, I think, but there will be at least some early data that we suspect Moderna and Pfizer are putting together, because they have been involved with trials since the middle of the summer. So we're hoping that in the next few months, we'll have a better idea. Now, even if we get to, say, a point where herd immunity is achieved, that doesn't mean the virus is gone. It just means that you don't have sustained chains of transmission. So there could be the opportunity, unfortunately, for sporadic outbreaks. And that's why I still think it's important for us to obviously open up the economy, get back out, especially if there's a decreased surge that we're seeing now, but still practice some degree of masking and distancing. The other concern that we have is that the variants, much about the variants is still unknown. We don't think that they are more transmissible, that is through the air, but we think that if they are transmissible, if they get to you, they may be more likely to be infectious. And so the issue of whether they're more transmissible versus more infectious versus more virulent are three issues that still need to be understood better. And we need to do those in observational studies. So I would be still very cautious. I would obviously want to still get back and socialize, but still with some distancing and masking at this point.
Great. One of your classmates from the class of 81 is curious about the issue of public resistance to receiving the COVID vaccine. And is that higher or lower than resistance of vaccines in general? And what do you think we can do in terms of policy-wise or education-wise to get people beyond the hump here about getting vaccinated?
Yeah, that's an important issue. And I'm a pediatrician, really. I'm doing a lot of these studies in adults, obviously, but as a pediatrician and a pediatric vaccinologist, boy, we've been called every name in the book. I chair this committee on infectious diseases for the American Academy of Pediatrics. It's called the Red Book Committee. We have a lot of anti-vaccination sentiment before COVID. I can see at this point from going to the CDC meetings pretty much at least once a month, if not twice a month now, that the tone of the public commentary has really shifted away from anti-vaccination to pro-vaccination, but also to more comments from the public about engaging trusted community leaders. And I think as providers, it is really important for us to lead the way and engage with our communities. That's really probably going to be the best way to get over this hesitancy issue. And frankly, a year ago, many of us were afraid, ourselves as experts in this area, about the way these vaccines might be rolled out, about the way the transparency of the protocols and the data. And it turns out that the companies themselves led the way in being very transparent with their protocols, with their data. The processes with the FDA and CDC have been untouched. They are normal, highly regulated processes. So we have that confidence, but we need to really educate our communities about that. And I think that's where some of the new hesitancy comes about, in addition to the usual mistrust of vaccines and government and pharma and all of those things that we hear. So engaging in community levels is going to be critical. The CDC has been really tasked with trying to help develop this. we received some NIH funding. I have an existing U54 NIH project to look at precision health and health disparities in communities of color that had nothing to do with COVID, but we were able to leverage that to build a supplement for working with indigenous communities here and communities of color here in the California area, as well as in indigenous Native American populations in the central US, where we already had existing relationships, to try to build really leadership within their own communities. So I think that's going to be critical, is getting messages out and into providers who really are just overwhelmed with treating patients and not really understanding a lot or having time to understand how these vaccines work and the safety signals that have been looked at and found to be really straightforward.
Terrific. So there's been a lot of talk in the recent weeks about quantities of vaccines and delaying the second shot, or the effectiveness of one dose versus two. Could you talk a little bit about that topic and your confidence in levels of production? And also, will news of vaccines like the J&J, which might not require a second dose or has less requirements in terms of temperature, help to relieve this concern?
Yeah, let me start with the last question first, because that's that one I can actually say I hate to say I don't know so much, but for that vaccine, the J&J vaccine, we actually participated in those trials here at Stanford. Obviously, there were many sites involved. That trial went very well. There were very few adverse events, at least from what we could understand from the national review of the data as the study was being rolled out. What we need to see is efficacy, and I believe that if they're coming to FDA soon, the efficacy must be pretty good. I don't know the data yet. And one of our faculty members here is also on the FERPAC committee, the FDA Vaccine Advisory Committee, and they don't have a date yet, but we understand that the company is ready. They shut the trial down right before Christmas. So they've had about a month to review the data internally, and I suspect they'll be submitting that data pretty quickly in the next week or two to the FDA. Once it gets to the FDA, they need to pour through the data. Obviously, it's a big data set with over 30,000 people enrolled as well. And then they will meet and we will be able to participate in those meetings to review the vaccine data for potential emergency use authorization. And as soon as that happens, within 24 to 48 hours, we will meet again with the CDC ACIP meeting group to look at indications for vaccination and dosing and intervals. So we're hoping that there should be another vaccine, the J&J Janssen collaborative vaccine, by the end of February, we hope. It is true that it's a single dose vaccine with no refrigeration requirements at this point. So we're very excited about the possibilities of getting that vaccine out to places, unlike Stanford, that don't have -70 freeze or other refrigeration. So we suspect that will help with distribution of the vaccine to more rural areas and areas that are less resourced. That should boost our supply. Now, coming back to the issue of doses, the answer is that there is no answer yet, and that came up this morning at the CDC meeting. Moderna and Pfizer are really looking at those data. It's very hard from an epidemiologic perspective to really extrapolate from, well, here was the interim efficacy between dose 1 and dose 2, because that's an epidemiologic measurement. What you really need to understand is the immunologic implications. That is, if you prime somebody with the first dose, do you need that boost of the second dose to really maintain that efficacy, or is a single dose enough? And that's a huge assumption to make. Most of us think that it's likely that a first dose could prime you enough to get you through a certain period of time. It clearly wouldn't be enough to get you to the same level that a boost would. And at this point, all of those immunologic studies are being done, and the epidemiologic studies as well. So at this time, there is no recommendation to just stick to a first dose. I know there's been a lot in the literature and in the public media, of course, social media, about let's just give everybody a one dose. We think it's better to give people their second dose and try not to deviate from the original recommendation. That said, if somebody can't get their dose right in the scheduled period of time, either the 3-week or the 4-week, depending on the product, as soon as possible. But again, it may be that that's not necessary. However, that's not the way the studies were done. So we'll learn more about that in the near future. And we just have to listen to the administration. You all probably heard that the new director of CDC yesterday, Rachel Walensky, said she did not herself know how many doses of vaccine there are available in the US. So we don't know how many there are, but we do know from the companies that production has not been a problem so far. So we do think there will be a steady stream of vaccine coming along. We just don't know how much that will be and how much we can amplify the distribution at this point.
And on the topic of production of vaccines, at this point, there is not a vaccine for children. What is the status for children in the development and in the outlook there?
So we also just got an update on that, and we are there are at least 4 trials, 4 vaccine products that are looking at trials in children: the Pfizer product, the Moderna, the AstraZeneca, and I believe the Johnson & Johnson as well. But none of them are any. The only product right now is that's a license for use in anybody under 18 is the Pfizer, which is licensed for use as young as 16 years of age. However, there were no individuals 16 years of age to 18 years of age who were enrolled in the trials. It was allowed, expanded use was, emergency use was allowed there. But all of the current trials there are trials now in 12 to 15-year-olds. There will be trials for younger children that are planned at this point. And the nature of the trials is a little bit of a patchwork. So some of the trials are going to be safety trials only. Some of them will be immunogenicity bridging studies, and some will be a combination. Now, you realize, of course, that safety trials require many, many more patients in order to pick up safety signals than, say, a straight immunogenicity trial. And I think one of the issues that Moderna and Pfizer are looking at right now are whether they can look at immune correlates of protection among the patients that I showed you in the table that were protected against infection compared to those who weren't protected, who were vaccine recipients compared to placebo recipients. Because if you could develop immunologic markers of immunity or correlates of immunity, you could then try to use those in trials for children, and then avoid the use of a full-on randomized controlled trial and just do a bridging study. So those we hope will be, some of them are in process now. Some of them are planned to be started in the spring or summer. So we think there might be potentially some vaccine indications for kids. We hope by the fall, but we just don't know at this point.
Can you talk a little bit about the after effects of COVID-19 and these long haulers, and what kind of research is being done on really the sustained effects of suffering from this virus?
Yeah, that's a great question. And there are a number of RFAs out from the NIH, from different institutes within the NIH to look at this issue of the long haulers. And obviously that the definition of long hauler is probably about a year, right? Because this virus hasn't been around much longer than that. So we really can't say much about what happens beyond, say, a few months to a year. There are trials going on at this time. We at Stanford do have one of our adult infectious disease colleagues here have a clinic that follows long-term individuals, including and immunologists as well. By the way, Kari Nadeau is following some of these patients to look at any immunologic markers that could, again, predict who might be at risk for these long haul symptoms. So what's really interesting and unfortunate as well is that when you look at these individuals, we don't really understand the prevalence. That is, how many people, what's the proportion of people who get sick who will have long-term symptoms? That's something else that is, we're trying to understand, given that we don't really have a great denominator. But one of the things that we've seen here in the clinic, and others have identified this as well, is that half of people who have long-term symptoms have what they call brain fog. So there's some degree of cognitive issues, and these actually turn out to be replicated in cognitive, and neurocognitive studies that have been done on these individuals. And the idea of looking at functional MRI or other markers is also being considered at this point as well. So those studies are ongoing, but it does appear that there are, as you have heard, a degree of people, a subset of people who do have long-term symptoms, and the neurologic and neurocognitive seem to be those that are the most concerning. And in some individuals, although probably rare, we do worry about the myocarditis that might also accompany some of these long-term effects. And there have been some early data from athletes and from children that suggests that there could be some long-term impact, but it's unclear what the degree and the nature of that would be.
Right. So there's been a lot of lessons that we've learned from this pandemic, and how we have approached it both nationally or worldwide. What are some of the lessons that we've learned that can help us when we face a crisis again, or things that we have learned that are going to help us in attacking other diseases like HIV or polio? Or so, what's the future outlook? Are we going to go through this scramble again if we have another pandemic worldwide? And what can we learn about public policy or handling a pandemic, collaborating across countries? And then what can we learn from this particular virus that will help us with other things such as HIV or polio?
Well, that's a great question. So I teach 2 classes every year. I teach global child health in the fall to the undergraduates, and then I teach epidemiology of infectious disease to the graduate students in the medical school, mostly masters in epi or PhD in epi students. And we were teaching that class last year, and I'm teaching it now. We're going to bring in Larry Brilliant to come and speak to us. Larry is an infectious disease epidemiologist. He actually is really one of these people who has been talking about this issue of pandemic preparedness for almost his entire career. For a popular culture reference, he was the consultant for the movie Contagion. I think was it Contagion? Yeah. He's just an amazing person. He's going to come and speak to our class, and I'm really excited to hear him talk about this. But clearly, I mean, you could read the tea leaves. We've been talking about this for a long time. So pandemic preparedness is something we've been doing for a long time, or talking about. The Obama administration literally had a pandemic playbook that I don't know if it was used or not, but they did have a playbook. I don't know if it was used or wasn't used enough, or whether we weren't ready for this virus. But we need to tune that up and make sure that it's really well resourced and well studied. I mean, even at our hospitals, and for those of you who work in a hospital setting or even in a clinic setting, we do tabletop exercises every year for preparedness. We should be more prepared for things like this. From the other perspective that I think we learn from this is that our public health infrastructure is pretty bad. And we, again, have been writing about, I trained at CDC after I left my fellowship and before I came to Stanford. And I have to say that the people there, I really considered staying at the CDC. Love the people that work there. They are amazing, really smart individuals, but they are working on shoestring budgets. They really don't have the infrastructure at the national level or even the local level to really make things happen in an efficient and a productive way. So I think that we've learned this pandemic has stripped bare all of the inequities that we already have and just laid them out for us to all see. The other area that I think we've learned about, although again have not solved, but at least we know it's there, are the areas of health disparities. This pandemic has really demonstrated how disparities can really magnify or amplify an epidemic. And by that, I mean that this really was a pandemic of inequity, that is, and people who didn't have access to care, didn't have access to proper PPE, to be able to isolate, to be able to work apart from others who in a safe way, to be able to access jobs that were not at-risk jobs, or even to have the at-risk jobs become less risky. All of those things played out in this pandemic, and we realized, I hope we realized, that that if we don't think about those things, they're going to impact us whether we want to ignore them or not. They will come back and drive a lot of what happens to everybody in this country, in this world. I happen to be a global child health researcher, and my work is in sub-Saharan Africa. I just found out two of my colleagues in Zimbabwe, one of them was actually an emeritus Stanford faculty, just passed away this week of COVID-19. These were healthcare workers and senior internationally respected faculty members who probably were infected due to lack of PPE, and I'm sure they don't have access to vaccine. So we need to really open up our academic and our intellectual and our scientific borders so that we can work collaboratively. And that is the one positive thing, that we have done a better job of that. I think with Zoom, in some ways it's, we are sick of it, but in some ways you can actually jump back and forth and talk to people across the world. So there's plenty of books and articles that will be written about this pandemic, but we have to learn how to be ready for the next one.
And my final question for you, Bonnie, is in the just couple of minutes that we have left, is tell us how your training at Stanford School of Medicine and your work in general at Stanford Medicine really prepared you for this last year.
Well, I have to say that I trained at Stanford, and then I went to Hopkins, and at a time, frankly, when we were not, known as a big clinical powerhouse. And when I went to Hopkins, people were surprised at how much I knew. And I thought, well, is that because I'm a woman, because I'm a person of color, or is it because I came from Stanford, or what is it? But the perception was that Stanford was, not a big clinical powerhouse. And of course, I believe that we truly are a world leader in clinical care as well as in research. But I learned a lot about the scientific method here. I did learn from fabulous clinicians here and how you translate that clinical, that scientific knowledge into the clinic. And I think that was one of the things that led me to public health and ultimately back to a public health research career here at Stanford. And more recently with this pandemic, I have to say that my colleagues at Stanford have been incredibly supportive. The whole community has come together. I don't think we've worked, I think we've been forced to work together, but that's actually been a wonderful opportunity to get to know people that otherwise we wouldn't have gotten to know. I'm sure that's happened with many of you as well. And everybody has just been in more than 100%, just working really hard as much as possible, whatever your background is, whatever your expertise is, on how can we deal with this, depending on your level of expertise. And the transdisciplinary, interdisciplinary approaches here at Stanford have been just amazing. And the supportive environment has been really what's gotten us through this so far.
Terrific. All right, I want to thank Bonnie Maldonado once again for this evening's presentation. A recording of today's seminar will be available on our website in the next couple of days. You'll also receive an email survey asking you about your experience in today's seminar. Please fill it out because your responses help us to serve you better in the future. And then join us again in March when our guests for the Alumni to Alumni Speaker Series will be Dr. Jeffrey Pollard and PhD graduate Dr. Anjali Shastri, who will be joining us for 23andMe to talk about medicine and genetics and some of the work they've also been doing on COVID-19 and genetic markers. Thanks again to Dr. Maldonado. Thanks again for all of you joining us, and good evening.